My name is Tela, and tonight we'll be reviewing aging's molecular effects on the brain's waste disposal mechanisms. In this presentation, we will be reviewing advances in research in proteasomal inhibition, autophagic lysosomal inhibition, specific protein interactions, and potential treatment mechanisms. Waste disposal mechanisms are crucial processes in maintaining the health and efficiency of cells. One of those ways waste is reduced is by proteasomes, barrel-like complexes that are key integrating damaged or misfolded proteins via ubiquitination. Other lysosomes are acidic organelles that break down waste products via autophagy. However, both of these functions decline with age, allowing the waste in the cells to accumulate and leading to major neurodegeneration. This major decay can develop into Alzheimer's disease and Parkinson's disease, among other neurodegenerative disorders, which leads into even more dysfunction. Developments on proteasomal inhibition origins provide insight on where and how it occurs. For example, 31 proteins display altered levels of expression in the hippocampus, parietal complex, and cerebrum in subjects with Alzheimer's disease. Those three regions express severe changes in their ability to function properly, such as in cellular development. The impaired cellular development leads to downregulated proteins and their ability, such as catalytic ability, which declines as age increases in subsets of memory and naive T cells. However, when USP14, a protein involved with weakened protein degradation, is inhibited, proteolysis in younger subjects cells increase, but not in older subject cells. Furthermore, older subjects' proteasomes show no decline in their proteolytic ability to degrade substrates. This implies proteolysis efficiency is not based solely on protein regulation. More likely that it's based on a variety of factors, such as internal defects and accumulated immunosubunits. Proteasomal inhibition itself has downstream effects that lead to negative consequences for the cell. For example, cell viability decreases significantly after 24 hours when exposed to MG115, a proteasome inhibitor. Following MG115's exposure, vesicle transportation ability weakens, and there's a significant decrease in the mitochondria. These decreases are some of the many precursors in early Alzheimer's disease pathogenesis, along with tau phosphorylation. Tau, an important protein for microtubule protection and stability, phosphorylates significantly at various epitopes when proteasomes are inhibited. This highlights the importance of proteasome functioning properly. When proteasomes are inhibited, MGRN1, an E3 ubiquitin ligase that normally acts in the cytosol, retreats into the cell's nucleus. This retreat allows for damaged or misfolded proteins to accumulate within the cell, which drives rapid neurodegeneration. New breakthroughs on autophagic lysosomal inhibition and its origins help gain more insight on aging's effects on the brain. AMBER1, a crucial autophagosome mediator, decreases over time in the hippocampus and neocortex. Significant decreases of AMBER1 in the hippocampal neurons make it difficult for the autophagic lysosomal system to function. Loss of function leaves the brain more susceptible to developing a neurodegenerative disorder. Neurodegenerative developments have been linked to C99 as well, a fragment derived from beta secretase. After being administered C99 enhancer, neurons displays exacerbated signs of autophagic lysosomal pathology, such as large vacuoles containing undigested materials. This allows for even more C99 fragments to accumulate within neurons, creating a harmful loop of neurodegeneration. When the autophagic lysosomal system is inhibited, severe issues such as ineffective digestion and accumulation of damaged or old materials occurs, such as proteins, lipids, or substrate fragments. Alzheimer's disease neurons show signs of immature autophagic vesicles, such as large vacuoles of non-digested waste and multimillar bodies. Also shown is excessive amounts of damaged mitochondria. Plaque buildup disrupts Golgi apparatus processes such as receiving lipids. Lipids such as cardiolipins are increased in the brains of mice with inhibited autophagy. Brains with high levels of accumulated lipids may have a relationship with Alzheimer's disease pathogenesis. Hyperphosphorylated tau, another molecule related to Alzheimer's disease pathogenesis, accumulates in the frontal cortex as seen in post-mortem human brain samples. These accumulations are hallmarks of neurodegenerative disorders such as Alzheimer's disease and Parkinson's disease. Downregulated proteins inhibit waste disposal mechanisms by weakening their ability to operate. For example, MGRN1 cytosolic levels decrease significantly with age. Despite limited functionality, MGRN1 is still able to exhibit a weak response for the neuron lysosomal system to react. However, this weak response is not enough in allowing protein aggregates, cytotoxins, and oxidative stress to accumulate until the cell perishes. Neuronal cells' waste disposal functionality and vitality also depend on their AMBER1 levels. AMBER1 is highly expressed in neurons at consistent levels necessary for related functions in autophagy. AMBER1, particularly in the hippocampus, decreases the older the subject is. However, increases in late age, particularly in the neocortex. This decline in resurgence supports the notion that unregulated autophagy is involved with senescence and inadvertently brings rapid neurodegeneration. Upregulated proteins disrupt cellular homeostasis and inhibit the brain's waste disposal mechanisms over time. For example, USP14's levels significantly increase with aging. These heightened levels, however, decrease the protein's overall effectiveness, thus allowing free ubiquitin cellular levels to rise, which leads to impaired proteasomal proteolysis. ERK1 and 2 is a regulatory kinase that phosphorylates tau and is implicated in Alzheimer's disease. It also is known to increase proteasomal inhibition. By inhibiting ERK1/2, this it rescues vesicle velocity, which has been reduced via this inhibition. Heat shock proteins such as HSP70 reverse aging's effects on the brain's waste disposal mechanisms. They provide protection and support in intracellular homeostasis. However, they decline with age, allowing neurodegeneration to occur. Heat shock proteins combat neurodegeneration in a number of ways. For example, EHSP70 increases synaptophysin levels, a crucial glycoprotein for synaptic vesicles. Synaptophysin levels are also known to decline with age inhibition. Neurons also accumulate lipofuscin, a lysosomal degradative residue, and neural age marker. Lipofuscin levels significantly decline following HSP70 treatment. This accumulation suggests that proteasomal and lysosomal dysfunctions are characteristic of neurodegeneration. Proteasomal activity increases significantly when treated with EHSP70. So finally, mice treated with EHSP70 experience longer lifespans compared to the control counterparts. This implies a longer-lasting waste disposal functionality. TTM saponin is a protein that counters aging's effects on autophagy. Its counteraction is facilitated via interacting with the Reb-mTOR signal pathway results. mTOR suppression via Reb decreases with age, blocking autophagy from occurring. Reb, a GTP regulatory protein, is upstream of mTOR, decreases with age. So TTM sapinin is able to increase Rheb expression, thus decreasing mTOR levels and restoring autophagy. Aged neurons also display increased levels of P62 and decreased levels of Beclin-1, two important autophagy-associated proteins. TTM sapinin restores these levels to their normal state. Developments on proteasomal inhibition show that the brain's protein levels and abilities decline with age, allowing neurodegeneration to occur. Waste accumulation and altered protein levels inhibit the autophagic lysosomal system, leading to neurodegeneration as well. Upregulated and downregulated proteins inhibit proteolysis, autophagic function, and disrupt proteolystasis. However, proteins such as HSP70 and TTM sapinin are capable of restoring the abilities of waste disposal mechanisms. A neuron's waste disposal mechanisms are at risk of decline due to aging. However, new developments in research allow for better understanding of how these dysfunctions occur, what they cause, and how to combat them. Thank you.